Dear students, this is ninth Biology Unit 18 Organization of Tissues. Unit 18 Organization of Tissues Part 8. Unit 18 Organization of Tissues Part 8. Last video class we have seen nervous tissues, and also we have seen the explanations of this basic structural functional unit of the nervous system is the neuron one, and also we have seen information bits, and we have started types of cell divisions. So we have completed a mitosis. Here these explanations we have seen. The next topic we are going to see is the one mitosis. Next topic we are going to see is the one mitosis. So what is mitosis? Mitosis means increasing number. The cell will undergo for multiplications. The cell which increase more, that is we call as a mitosis. Here this mitosis was first discovered by Fleming in 1878. Sorry, in 1879. In this cell divisions, one parent cell divides into two identical daughter cells. One parent cell divided into two identical daughter cells, each with a nucleus having a same amount of DNA. So that is mitosis. We'll see the diagram of the mitosis here. The picture what you are seeing is the one. This is the events of mitosis, the stage which occurs, the process of mitosis. You can find here, is there in your textbook. Here, the first one, maternal, that is the mother cells. Paternal means father cells, we can see here. We'll see the stages of mitosis. The stages of mitosis. First one, this is we call as a interphase. This is we call as a interphase. I'll tell you the diagram, events one by one. The next one, prophase one, and the next one, metaphase. So this is the one, prophase, and metaphase one, and anaphase one, and telophase one. So these are the events of mitosis. Now we'll see here, as we have seen in cell divisions, one parent cell divides into two identical daughter cells, that each with a nucleus having same amount of DNA and same number of chromosomes and genes as a parent cells. It is called as a equational divisions. So they divide equally, that nucleus as well as DNA as well as gene. That is we call as equational divisions. The mitosis consists of two events. Mitosis consists of two events. They are karyokinesis and cytokinesis. What is karyokinesis? The divisions of nucleus into two daughter nuclei is called karyokinesis. We'll see all these things. Then what is cytokinesis? The divisions of cytoplasms, that is we call as a cytokinesis. So all these things we'll see here. First phase is events of mitosis is the one. First phase is the interphase. As you have seen the picture here, this is we call as a interphase. You can find this diagram. So it is the resting phase of the nucleus. The nucleus undergo for the resting one. That is why it is called as the resting phase of nucleus. It is the interval between two successive cell divisions. It is the interval between two successive cell divisions. The cell prepares itself for next cell divisions. The cell prepare itself for next cell division. That is we call as a interphase. Okay. The next one is the one karyokinesis. Next one is the one karyokinesis. I'll just tell you now. We'll see. You can take your textbook here. So karyokinesis. This is a divisions of nucleus into two daughter nuclei. It's called karyokinesis. You can mark it out. 2 marks or 1 word also will ask you. It consists of 4 phases. Consists of 4 phases. They are prophase 1, metaphase 1, anaphase 1, telophase 1. There are 4 phases are there. That is prophase, metaphase, anaphase, telophase. We'll see one by one what it is. What is prophase? Pro means first. During this stage, the chromosome becomes short and thick and are clearly visible inside the nucleus. You can find, we'll see the diagram here. First phase, we'll see the karyokinesis. They undergo 4 phases. That is, as we have seen, prophase 1, metaphase 1. The karyokinesis, they undergo for the 4 phase. First one is a prophase, second one is a metaphase, third one is anaphase, last one is the one telophase. Now, first we'll see prophase 1. Pro means first. During this stage, the chromosome becomes short and thick. During this phase, the chromosome short and thick and are clearly visible inside the nucleus. Can you find? Then inside the nucleus, it can be clearly visible one. Okay, and the centrosomes split into centrioles. The centrosomes split into centriole and occupy the opposite poles of the cell. The center one we call as centrioles. Okay, they occupy the opposite pole of the cells. Each centriole surrounded by aster rays. The aster rays, each poles are surrounded by aster rays. The spindle fibers appear between two centrioles. The dot-like structures here. This is because the spindle fibers where they are going to arise. That's what they say. Here, spindle fibers appears between two centrioles. Spindle fibers appears between two centrioles. The nuclear membranes and nucleolus disappear gradually. The nuclear membrane and nucleolus disappear slowly or step by step. The next one, metaphase 1. Next stage is of karyokinesis. Second stage of karyokinesis is the one metaphase 1. Here, this is the diagram. What you are seeing is the one metaphase. Metaphase. Okay, here meta means after. Meta means after. The duplicate chromosomes arrange on the equatorial plane. Equatorial means the center part of that nucleus. That all the chromosomes should be arranged as equatorial plane and form metaphase plate. Each chromosomes get attached to a spindle fibers by each centromere here. Which is what is spindle fibers? Here the dot are like structures which arises from on top also from the bottom also they arises and the top also they arises. That is because of spindle fibers. Each chromosomes gets attached to the spindle fibers by its centromere. Centromere means the white color dot like structure. This is we are centromeres. In the centromeres only the spindle fibers they go and attach. The centromere of each chromosomes divide into two by being associated with the chromatid. A chromatid, it's the help of the centromere as well as chromatid, they divide into two. So this is the information about metaphase. The next one is the one anaphase. Here, ana means up or back. The centromeres attaching two chromatids divide and two daughter chromatids of each chromosome separate and migrate towards the two opposite pole. Okay, here the centromeres attaching and the two chromatids just divide. Can you find that centromeres versus chromatid? They're just going opposite directions. They're dividing it. And the centromeres attaching the two chromatids divide and the two daughter chromatids of each chromosome separate and migrate towards the opposite pole. Okay, can you find they're just migrating, they're moving towards the opposite pole. of that spindle fibers. There's a spindle fibers just pulling out by attaching the centrum here, and just the dividing, moving opposite pole. The next phase is the one telophase. Tele means end. Here, each chromatids or each daughter chromosomes lengthen and become a thinner one and become thinner and turns into a network of chromatin threads. Turns into a network of chromatin threads. Spindle fibers break down and disappear. Spindle fibers break down and disappears in each daughter nucleus. So this is what the information is about the telophase. So this is what the events of mitosis. So there are four phases are there. Karyokinesis means divisions of nucleus into daughter nuclei called as a karyokinesis. While they divide nucleus into next one, they undergo for the four phases. One is the one prophase, metaphase, anaphase, and telophase. This is what we are seeing so far. Okay, the next topic is the one cytokinesis. Here, this is what the cytokinesis. The divisions of the cytoplasm into two daughter nucleus. The divisions of cytoplasm into two daughter cells by constructions of the cell membrane is called as cytokinesis. The cytoplasm they just divide into two by constructions of the cell membrane. That is we call as cytokinesis. Just mark it out. 2 marks. Okay. And next one, significance of mitosis. What are the importance of mitosis? The equational divisions result in the productions of diploid daughter cells. That is 2n. Equational divisions result in the productions of diploid daughter cells. Diploid means two. 2n means two number of chromosomes here. That's what say. With equal distributions of genetic materials. Okay, and while they divide into two, they divide equal distributions of genetic material DNA. Okay, and second one significance of mitosis is the multicellular organisms growth, organ developments, and increase in body size are accomplished through the process of mitosis. In multicellular organism, growth as well as organ development and increase the body size. These are done by through the increasing of this process of mitosis. Okay, the next one, mitosis helps in repair of damaged and wounded tissue by renewal of the lost cells. If you got any hurt, if you get any hurt or wound, nearby wound, the cells undergo further divisions. By that divisions only we call as a repeated of process of mitosis. With help of mitosis, the repair damaged tissue. Damage means that wounded tissue, affected tissues, are again coming back by these divisions. That is because of mitosis. So these are the significance of mitosis. This is important. 4 marks can be asked. Okay, this important 4 marks can be asked. The next topic is the one meiosis. What is meiosis? The term meiosis was coined by Farmer 1905. Scientists call us a farmer, underline. It is a kind of cell divisions that produce the sex cells or the gametes. So meiosis was coined by Farmer 1905 is the scientist one. The kind of cell divisions that produce the sex cells or the gametes. Here the meiosis they produce only the sex cells, the gametes. Male is the one sperm, female is the one egg. Whereas mitosis, the somatic cells will be increased number of more in number by the process of mitosis. So that is the two difference between these two, mitosis and mitosis increasing number. A single cells they increase two and just they go on. Whereas meiosis, the reduction division takes place. They form sex cells or gametes. It is also called as a reduction divisions. Mitosis increased divisions, whereas meiosis reduction division. Now number will become half of number. That is why it is also reduction divisions, because the chromosome number is reducing to applied. The reason is the reduction division because the chromosome number is reduced to applied. That is n, 1n from diploid to 2n. For example, we have 46 chromosomes. Human being, they have 46 chromosomes. When it passes on to the next generations as a gametes, means the female as well as male, they have half number of chromosomes. 46 chromosome means 23 chromosomes from male, 23 chromosomes from female through the sperm and egg. That's what they say. The reduction division, because the chromosome number is reduced to applied. As a 1n, that's we call as a applied from diploid to n. They are converting 2n2 into single one, single n. That's what they say here. The meiosis produce 4 daughter cells from the parent cell. The meiosis produce 4 daughter cells from a parent cells. Okay, this is what the information so far we have seen. Today we have seen what is meiosis, introductions we have seen, what is the significance of mitosis we have seen, and also we have seen what is cytokinesis, and also we have seen what is karyokinesis and its types. They'll ask you 7 marks questions. Explain the event of mitosis. So they'll also ask question like this. And indirect questions can be asked like this. Explain the process of types of process or types of karyokinesis. They'll ask you. Both are same question, same answer only. Go through. If you have any doubt, you can contact me through WhatsApp. Okay, bye. Take care.